All right, welcome back, guys. So last time, in the last clip, we looked at transcription going from DNA to making RNA. So today we're going to look at the second piece of this. We're looking at how that RNA is processed before it can leave the nucleus and travel to the cytoplasm to find the ribosome. And then once it's there, what the ribosome does in terms of uh, manufacturing a protein from that information. So the first things first, we're going to go ahead and start with that mRNA that we had from before that was just produced in the nucleus by the RNA polymerase. Here it is. Um, and what we'll do is we're going to bring in this animation again. And we're going to, if we go all the way back to a gene here, we're going to do that. We're going to work backwards and we're going to look at this gene. So this gene, not all this information is for just one protein. We could have information here for several different proteins. It could be dozens of proteins, in fact. There's also information in here that controls how long a protein is able to be transcribed. So how long that mRNA exists in the cytoplasm, along with the amount and the quantity that's produced of that mRNA. So there's going to be information here that's important for coding the protein, but there's also information in here that is important for other processes. So if we roll this forward, we call those things exons and introns. So the exons will be the important coding information, and the introns will be the information that serves other purposes, for like making small interfering RNAs or microRNAs. So what we're going to do is we're going to go back to where we draw here, and I'm going to break this up. I'm going to break this up into, we'll say exon 1, we'll say exon 2, and then we'll just roll with that. Exon 1 and exon 2, and we'll say that I've got intron 1 and intron 2. The numbers aren't really important. All right, and again, this is my messenger RNA, my mRNA. So let's just get some definitions up here for you. So an exon, so an EXON, an exon. An exon is a section of DNA or RNA that is going to code, so it codes for proteins. So it's a section of DNA or RNA that codes for proteins. That's the information that actually is going to code for a protein, right? And it could be, so we could have, this could be a protein product, exon 1. Exon 2 could be a different protein product. Exon 1 and 2 together could be a protein product. And if we had other exons, like 3, 4, 5, and 6, it could be a whole host of proteins from that one gene, okay? The introns then serve other purposes. And it's not just junk. At one point in time, it was thought as junk DNA. That's not the case. They serve critical regulatory functions. So these are sections of DNA or RNA that regulate, so it controls the timing, when, how much, and the amount, essentially, of gene expression going from an mRNA to a protein that takes place. Right? And it does this by really managing how long that mRNA is allowed to exist out in the cytoplasm. All right. So in order to get just the information that codes for proteins into a molecule that a ribosome can use, we're going to have to cut out those introns or cut out the exons that aren't being utilized at this moment in time. And we do that by a process called splicing. Splicing. In splicing, we're going to cut out the introns. Cut out the introns. And we do that with a pair of essentially cellular scissors. And that cellular scissors is called a spliceosome. Spliceosome. They're like a pair of cellular scissors. So what's going to happen is we're going to snip out the introns, and what we're going to be left with are just the exons. So after that process happens, we're going to still have our mRNA transcript. So I'm going to redraw that. Here's our mRNA transcript, 5 prime, 3 prime. And actually, I've kind of misdrawn this. This shouldn't be that large probably, but I'm just going to have exons. So I might have like, this might be exon 1, and this might be exon 2. Right, and the introns then were cut out. Before this is allowed to travel out into the cytoplasm, I want to protect it. So I'm going to put a 5 prime, what's called a 5 prime cap here. And typically that's going to be like a guanine. And that's going to protect that end from endonucleases that might denature the RNA. And it's going to also allow for the ribosome to locate and attach to this. I'm also then going to put on a bunch of adenines. It's called a poly-A tail, a polyadenylated tail. Okay, and that's going to then protect that 3 prime end. This process of cutting out introns and piecing together the exons that are important, it's called splicing, but there's something called alternative splicing. Alternative splicing. And it's this idea that exon 1 could be by itself and produce a protein product. Exon 2 could be by itself and produce a protein product. And exons 1 and 2 could be by together and produce a different protein product. So from this one gene, we can have 3 different protein products. So alternative splicing, it provides an advantage to cells Go and grab it. No idea, man. Okay. I don't know where it was gone. Okay. Sorry about that. Somebody interrupt. Because it's so an advantage because we can make lots of proteins with less DNA, right? So we can have a smaller amount of DNA and genes and produce a lot of different protein products, right? There's a huge demand for all these different types of machines and protein products that a cell has to make. And the instructions have to all be contained within that DNA. So it provides us a way to minimize the amount of DNA, that burden, and yet produce a lot of different protein products. So think about like a desktop on your computer, right? And you have all these different documents and PDFs and what have you. And you can put those things in folders, and then minimizes the amount of space that it occupies on your desktop. Okay. So think about if you didn't have folders, you just had all these documents all over the place, right? It would take a really long time to sort through and figure out and organize. So for a cell, being able to store the instructions to make these proteins in a limited amount of genes and not, exponentially more genes is a space saver and just a more efficient way of doing things. So we can look at this. We'll pull this up on this clip, right? So here we have the introns, we have the exons. We're going to go ahead and cut out those introns with the spliceosome. So we're going to see that here. Spliceosome is going to come in here. It's going to pinch together the intron and snip it out. And then it's just going to glue together, ligate together the exons. Right, and you can see there the 5' cap, the polyadenylated tail, and this is going to travel out into the cytoplasm and it's going to find a ribosome where the next process, translation, is going to take place. So let's go ahead and talk about that briefly. So we're going to look at what's called translation. So translation. Okay, so in translation, we're going to have the ribosome is going to locate and read that mRNA. So the ribosome is going to locate and read the mRNA, And then once it does that, it's going to assemble the amino acids that make the protein, that comprise the protein. And we'll sketch this out, but I need a new piece of paper to do that. So translation is the process of that mRNA being read by the ribosome and assembling the amino acids in the right order. So let's just kind of walk through this and then we'll check it out in the animation. We'll call it a day. So here is my RNA transcript from before. 5 prime, 3 prime. Again, that's the location of the nucleotides. Just gives us some orientation. Here's the 5 prime cap. Here's the polyadenylated tail. And what the ribosome is looking for, it's going to use this 5 prime cap to help somewhat identify where the mRNA is at. And then it's going to look for a start codon. So it's going to look for like a start code. Start code. And the start codon is what we call the codon AUG. So they're nucleotides with the nitrogenous bases adenine, uracil, guanine in succession. AUG. All right. And the ribosome kind of looks like this. It's 2 units. We have a large subunit and a small subunit. Okay, ribosome. Large subunit and a small subunit. And it's going to be looking for that start codon. So maybe here's the start codon AUG. Right, so it's going to start working this direction, reading the codon, and bringing in these little transporters called tRNAs. So here's kind of my tRNA. So this is a tRNA. And attached to the top of the tRNA is going to be the corresponding amino acid. So in this case, it's methionine. So it's going to be traveling to the ribosome there. It's going to bring that amino acid in and form a peptide bond with the next amino acid as it reads the next set of codons. So maybe the next set of codons is like GGG. So then whatever amino acid corresponds to that codon would be that next amino acid in the sequence. And we'll see this here in the animation. So again, we want to define what codons are. A codon is a set of 3 bases. So a set of 3 bases that codes for an amino acid. And we're going to see that there's 64 codes and 20 amino acids. So there's redundancy in the code, which is an advantage for a cell because it minimizes the impact of mutations, especially to that 3rd nucleotide in the codon. So let's go ahead and bring in the animation here and hopefully provide a good framework for you. So here's that mRNA. And we can see the multicolored nucleotides here with the multicolored nitrogenous bases. So there's GCA. So what we're going to do is look at this table, right? So 64 codes, 20 amino acids. So GCA, we would look at G in the first position. That puts us in this horizontal row. Then we look at C in the second position, would put us in this vertical column. So where the horizontal row and the vertical column meet is right here in this box. Then we bring over that last nucleotide, A, in the third position, and that gives us GCA. We can see GCA right here. So that codes for the amino acid alanine. So if we go back, that GCA is going to code for the amino acid alanine. So we would know when we bring the RNA back in the picture that GCA would be for alanine. There's AUG, our start codon. So let's go ahead and look at that table again. AUG, A in this horizontal row, U in this vertical column, that puts us here, and then G, bring over the third position nucleotide. So that is methionine. So we have AUG, that's our start codon, that is methionine. So the amino acid that's going to eventually reside in that spot, over that spot, is going to be methionine. And then you saw the stop codons there as well. So there's our our small ribosomal subunit, there's a tRNA with the amino acid attached to the top. We call that a charged tRNA. At the base of that tRNA, you see an anticodon. That's going to correspond to the codon on the RNA. So it's going to come in, it's going to hydrogen bond. So we're looking at the codon AUG, hydrogen bond with the anticodon UAC, and then up top there, you're going to have the amino acid methionine. So we have a small subunit. We're going to then bring in the large subunit. It's going to come sit down. And you'll see the subunit's got 3 different positions. It's got an E site where the tRNAs are going to exit, right here. It's got a P site where a covalent peptide bond is going to form between neighboring amino acids. And then it's got an A site where the next tRNA is going to arrive. So E, P, and A. Exit, peptide, arrival. Easiest way to remember that. So we're going to read the next codon. If it's GGG, this would be CCC. We're going to need hydrogen bonding here. We're going to get a covalent peptide bond between the amino acids. And then this complex is going to slide down. We're going to get bonding here. We need the peptide bond that forms. This is going to slide down. Our tRNA is going to leave. So our tRNA is going to exit now that it's uncharged and go pick up another methionine in this case. And then the next one will come in. Hydrogen bond. Get the peptide bond between the neighboring amino acids. And this is going to continue. It's going to continue until we reach one of those stop codons, UGA, UAA, or UGA. Right? So there's our stop codon. And it corresponds to, if we go back and look at this table, let's see, table is going to be right here. So UGA, UAG, UAA. And it corresponds to what we call a release factor. There's going to be a release factor there. So this release factor then is going to disassemble this complex, and we're left with this protein, right? This protein is going to form into a 3-dimensional shape based on the properties of the R groups on the amino acids and based on how neighboring amino acids in different parts of the chain associate, whether it makes an alpha helix or a beta sheet. It's going to fold up into that secondary, tertiary, and quaternary structure. And then that structure is going to dictate what it does, what its function is. If this is a protein to be exported, it's going to be manufactured on ribosomes that are on the ER, the part of the endomembrane system. They're going to travel in a vesicle to the Golgi apparatus on the cis side of the Golgi here. And then they're going to be modified and repackaged and leave the Golgi on the trans side of the Golgi, travel to the plasma membrane, which we'll see here. All right, so we're going to travel away from the Golgi to the plasma membrane, exocytosed, and then released into, in this case, it's the lumen of the small intestine. And these proteins are helping digest these materials that this person had eaten. Okay, so that's the full process of transcription, translation, central dogma. We'll do some work with it in another session later this week. So I hope that's helpful, and I'll see you next time.